Great. So, good morning, good afternoon, good evening, good whatever time zone you're on. It's lovely to see you all, and welcome to Coffee Buddies at Biotech Gate. And as yesterday I said, and I will repeat myself, some years ago, Graham and I bought a virtual Citroen CV pop-up coffee car, and we drive around and have been driving around digital and real life events for some time. What we really like to do, our major interest is to engage, inform and entertain people in the life science sector. And as I said yesterday, the life science sector has exploded. So almost anything is life science now, between smart clothing to smart watches to drugs to to synthetic biology. So we get a hugely heterogeneous group of people together, which makes our time together extremely interesting. And today we've got another very interesting heterogeneous group of people who are covering all sorts of things. And what we try and do is bring a little bit of serendipity to digital events when we do this. And in our real life events, and we run CEO for and other events, we try to bring people together to share information, what's going on, who's the contacts, where's business to be found. And by and large, we've been very effective at helping people grow their businesses. And as a substrate to get our juices flowing and our thoughts together, we like to invite interesting people to come and talk to us. And yesterday we invited Eric, who's going to talk to us as like part two of Eric, but a different topic today. Part two of Eric, but different topic, to catalyze our thoughts. And I'll introduce Eric and say, Eric, how are you? And for those who weren't here yesterday, maybe you could just introduce yourself, say where you are and what you do.
Yeah, Tony, thank you. And it's good to be back. I've had a long career in big pharma, probably 25 plus years, most recently in Singapore, about 5 years, starting up a translational medicine research informatics team in Singapore for MSD. I left that role about a year ago and have started working with CDD Vault, Collaborative Drug Discovery. And I'm happy to share with you some of the details of the project. And then more importantly, I think for this discussion, and I really would like it to be a fairly interactive discussion, the topic of artificial intelligence, machine learning, computational sciences in drug discovery. I'll tell you my area of research. So I'm a PhD computational scientist, data scientist as we're called today, from the very beginning, starting out with, Hansch, Free Wilson, QSAR type models, all the way rolling through, recursive partitioning, support vector machines into deep learning, primarily in the research space, but more recently in genomics, genetics, and real world evidence space, trying to make sense of all that data and advance human health in the context of mainly large pharma.
And I think this is something that is incredibly close to all of our hearts. What is this, how do I describe it, emerging yet transforming way that we conduct our business. So maybe you could elaborate, Eric, what your thoughts are in the AI machine learning space.
Yeah, I think it's quite obvious there have been tremendous advances, right? The rate of change is I think never been experienced like this before, right? I mean, when I think about how I started, a short 25 years ago, where we had machine rooms and, we're doing linear regression, SAS, statistical models, still all very important, don't get me wrong, that is incredibly important. But the advances that have been made and the types of things that we can model and the data sets that are available to us to enable the artificial intelligence approaches are unreal, right? I think the biggest thing that we have to watch out for is data quality. It's been that way forever, right? There used to be an old adage, garbage in, garbage out. I think that's, just because we have more data doesn't necessarily mean that we're going to get better models. But I think data curation, right, management of data, comparability of data, all incredibly important components to building a successful AI program within a pharmaceutical company, all the way from target identification out to post-marketing, drug safety surveillance, and frankly, even into manufacturing.
And I suppose the hypothesis here is that you can make sense of a big mess. You know, so the hypothesis is, I mean, I've worked in clinical for years, and you know, you used to look at the data and you just couldn't work it out until some biostatistician started doing his magic. And I suppose the question everybody's trying to understand is, who's got the sort of data that we want to explore? Is it, I mean, there's clearly public data. There's clearly a lot of private data that you just can't get your hands on. And then there are different levels of data dependent if you're a small biotech, which maybe only has one notebook full of data, compared to, say, Roche, which has close to 150 years of data. So I suppose with that background, what's your thoughts, Eric?
Yeah, I mean, this is exactly the key question, right? And I think there's one other aspect to this is the data, the heterogeneity of the data, right? Structured versus unstructured data. All those things come into play. But you know what we always did, and I still think it's useful, is a fit for purpose model. What is it that you're trying to predict, and how good does your prediction have to be to actually get you closer to the goal line, right? And so, you know, to address your question, if you're at those final stages and trying to refine, let's just say a compound interacting with a protein, for example, in the crystal structure, then you need that high level resolution of interaction data points. Yeah, that's totally different than if you want to do modeling of, let's say, a high throughput screening experiment, right? Where you're just trying to get the kernels, the good compounds, the potentially good compounds out of the millions and millions of compounds that you could have selected, right? And at that point, it becomes increasing your odds of success, right? It's a probabilities game. You know, I often put this in the context of a casino, right? If I could just be a little bit better, if I just beat the house odds a little bit, you know, I could be quite successful, right? But I don't need to be perfect every time. I just need to beat the house odds. And that's where, you know, it's not a game, but I think that's one way that we can sort of think about how these models should be interpreted. Now, there are other areas where we have to be incredibly accurate, right? I mean, think about self-driving cars, for example. I know this is a topic for drug discovery, but, you know, would you really get into a self-driving car if you didn't trust that vehicle, right? And, you know, we're getting there. There was a time when autopilot was a new thing, right? Nobody trusted an autopilot. I need that pilot with hands-on controls for the entire flight, right? But I think we've all gotten to the point where we sort of trust autopilot. I think, you know, drug discovery, there are other things to think about, right? Because patients' lives matter. Patient safety is a very serious concern, right? And so that's actually one of the areas that I've been focusing on, is how can we use this data all the way from genetics to real world evidence to improve drug safety through a variety of different modeling methods. And we have to be really good when it comes to that type of
thing.
So I was wondering whether, Eric, you had some slides you wanted to show, or how do you want to conduct this?
We can go either way on this. Let's
just ask a couple more questions, and then we'll bring a couple of slides in. I mean, I think there is a, from what I'm hearing, and I'm interested in your comment, that, I mean, I spoke to someone this morning who said they were telling me a story of a person who joined a company, and since that person joined the company, I think it was 14 people have left. And I said to that person, well, this is associative, not causative. So my question, what I hear, you know, on the grapevine, the big breakthroughs are going to be when we can work out causative. Now, I've spent ages reading and listening to Judah Pearl, but is the causative next generation causative machine learning and AI the way forward? And will it come to a point where I won't be getting a lot of false negatives?
Let's hope so, right? I think I don't really want to confuse the issue of just necessarily false negatives or false positives to causality, right? I think you can be, correct in a prediction, right? You can reduce false positives, false negatives, and not really have causality, right? I think it's incredibly important to understand the causality, right? That's what we all strive for, is that deep understanding of the biological underpinnings of human health, right? Whether it be genomics, genetics, protein-protein interactions, networks of interactions of different things, right? And then you put the time component on that and the concentration, and then you put in environmental factors, right? So it's an incredibly complex problem. And to understand the causality when you have so many different effects on a system, very difficult, right? So I think in the short term, in many times we're just happy to get the association that seems logical, we accept as biologically reasonable. However, it may not actually be a causation that we're observing. But having said that, it's useful at the very early stages, the research discovery stages, to guide a program. And what we always thought about was not so much getting the real understanding, right, of the underpinnings of the biology. Yes, that would be nice, but that's actually an academic problem, and we partner with a lot of academics to get at that. What we were looking to do was propose the next experiment, right? What is the hypothesis that we can now test in the laboratory, right? And use these models in an iterative way to build up hypothesis, disprove a hypothesis, guide the evolution of our project towards some endpoint based on what we know. And hopefully, and I think it's reality, computers can help us do that in a much more efficient way, especially with the data management piece, right? Especially, it's very difficult to do things when you have isolated spreadsheets floating around, right? And when you mention people leaving, X number of people leaving over some short period of time, the first thing I thought about is, oh my goodness, what happens to the data, right? What happens to that brain power that just walked out the door? Did they leave anything behind for the company, right? And that's where I think, and here comes the plug, right? The shameless plug for CDD. But that's where I think CDD and other systems become incredibly important for capturing all of that information, right? And keeping a history of all that has gone before you, because I may or may not be at a company tomorrow for whatever reason, but the information, the value that I contribute to the company should remain beyond my
Exactly. So Eric, have you got a couple of slides that you can bring in?
I do, and we can share those if you like. I'm going to go through, and forgive me if this is repetitive. I do, I'm doing some work for CDD, so I feel obliged to talk about, and I actually believe in the product quite a lot. So let me talk to you a little bit about the CDD Vault, Collaborative Drug Discovery, which is a company, by the way, that I joined about 6 months ago. And some of my colleagues are on the phone. If you have some detailed questions, I look to them to jump in to answer those types of questions. But what CDD provides is an integrated system, a 4-component system, where it allows you to register both your compounds, your entities, it could be biologics, your experimental data, right? Organize your experiments, conduct experiments, propose experiments through an electronic lab notebook, track the inventory of those entities that you register, right? And then a very nice visualization tool to help make sense of it all, right? And so that's great from a user, that's from a functionality point of view. But what I'd like to talk about today is that we really have a system that's set up for dual purpose. One is the scientist in the laboratory, right? And by scientist, I don't mean the data scientist. I mean the bench chemist or the biologist who really doesn't want to be bothered with all the informatics, right? The old phrase is, don't make me think. Don't make me learn yet another software system to get my work done. Just provide me an easy to use tool. And I think CDD Vault has actually done that. It's very well established in terms of the bench scientists and their ability to put in and extract out data and meaningful hypotheses. But what we talk about today, which really enables the artificial intelligence work, is the API, right? The application programming interface capability to the Vault, which can be accessed. And I've just forgive these logo soups, as I call them, but here are all the partners that work with CDD. There are many more. I've just selected some that provide connectors to CDD and allow, through a programmatic interface, the ability to access the data that the biologists, that the chemists enter into Vault. You can pull that data back out, put it into your favorite modeling system, right? Build AI models to your heart's content, whatever it is, and then beautifully push that data back to the Vault so that the scientist can access the models, right? That's been one of the biggest gaps in my experience in this entire industry. Is you'll have data scientists in one part of the organization building models, right? Very predictive models. They always love the models. They don't love, they think they're the best models. And then you have all the scientists. And how do the scientists actually interact with the data scientists to use those models, right? Or the predictions from those models. And I see CDD as Vault as providing that access, right? So we'll stop here. I don't know if there are any questions about CDD or if that prompts any thoughts, and then we can go to the.
We'll take a few questions from the audience. I think if we can bring the slides down.
Yes.
I think let's just take some general questions before we go into networking. I mean,
okay.
Just looking at Denise and our colleagues at Allen Advantage, this is very much your space that we're talking about. Have you got any thoughts that you'd like to add to us?
So we were listening to Eric's answer in terms of the applications of ML technologies here, and we pretty much concur with what he just said there.
Okay. Okay, so other comments around the table?
Kaz, you're a scientist, as it were.
Yeah,
Tom, yeah.
Yeah, hi Eric. As a scientist, and background over many years, I think I mentioned to one of your colleagues yesterday, I set up a Heath Robinson system similar to yours in the 90s. We had great California software. We had 64-bit supercomputers, and no hard lines, to connect it all. So it was chemists finding biology data, et cetera, and trying to build it up into the clinical side. But your point about the cause and looking at the cause as a sort of philosophical way of saying this is the cause, and using supercomputers to analyze all this complex data, to bring it down. And I had a slide, many years ago, I think it was a Tolstoy quote from, War and Peace, where basically it said, no one thing is the cause. All this is only the coincidence of conditions under which every organic event of life is accomplished. Now, essentially that's what you're saying. You've got all these complexities, but you're saying this is the cause, or this is the drug, or this is the receptor, or this is the protein, et cetera, et cetera.
I mean, Tom, it's a great point, and think about, and I just have to throw out there, I don't know if you remember Evans and Sutherland and dial boxes, and looking at the dark rooms for looking at protein structure interactions and things. So yeah, I was there. I was part of that generation. But back to your point about the cause, it's really, as you said, difficult to get to that, and it is incredibly multifactorial, right? And environment. I think one of the things that I picked up over time is that, everyone's different, and that sounds like, yeah, of course, right? But it really is true, right? And what a big component genetics and epigenetics plays in patient populations. And when I was, I spent the majority of my career in the U.S. and worked with a lot of U.S. databases, right? And then kept thinking about, well, what happens when we go to Asia and the patient populations? And there's another aspect, there's a very important aspect to this. And I don't want to get caught up in buzzwords and whatnot, but the diversity and inclusiveness of our clinical trials and how certain medicines or even digital health solutions will work better in one population or one group of people than another group of people. And how do you tease apart those factors when, in fact, I, as a biologist or a chemist, PhD trained, I'm only looking at the data but not considering, the sources or the environment of the individuals, right, or the system from which that data was generated. So I think there's a lot of undocumented parameters that we really should consider, and hopefully AI can tie that all together for us, right?
Yeah. Well, I think it is a hope, and we're certainly seeing some evidence from AI, to suggest that we can go into these epigenetic, phenomena and, personalized medicine.
Yes.
Okay, thanks, Eric.
Just wondering if there's any other questions. You just put your hand up if you've got a question at all, or if you've thought of one. It sounds like, looks like everything's been very well covered. Yes, I think over to you, Tony.
Graham always does this to me. I mean, we can have a long academic conversation here, which I think I would love to have, and Tom, I'm sure you would love to have. But this idea of I think the way that it's going to be is that you will have a department of AI in your company, and they will produce you a stream of potential structures, and then you will have other ways of doing it, and it's going to be just part of the mix. And I think that what people tell me, and Judah Pearl seems to be the man to read, but I can't digest his stuff, is that there will come a time where you can almost ask an open question. What protein is inducing this rheumatoid arthritis? And there will come a time where we'll be able to ask those why questions.
Yeah.
Now,
Tony,
that's mind-blowing.
Yeah, Tony, I did a thought experiment. This is a really old and well-documented experiment, but I actually played with Google again today. And there's this notion that if you put in woman or CEO, what are the images that come back for a CEO? Very few women, right? If you put in model, all female come back, right? So, I share your optimism. I hope someday that we can say why through some sort of magical one box, pose a question, but until we can, really get some of these search technologies better and less biased, if you will, I think, there's a long road ahead of us in that context. So I'm hopeful, but let's hope I live to see that day.
I mean, there is a lot of literature about so much redundancy in systems that Tom, that you can go up a blind alley quickly.
Yeah, we talked about this, yesterday with Renate, with regard to legacy data. There's so much, as you say, somebody comes into an organization and so many people leave, and, how do you troll that sort of data, when there's nobody there to point you in the right direction? And, the sad thing is, pharma, big pharma, is one of the main problems. But the future, I think, is, these young startup biotech companies, because they're more sophisticated, they're more in tune, they've got the technology which allows them, to move faster. And also, hopefully, the data storage is far better than some of the legacy data that you see in big pharma. And obviously, this is where CDD Vault and others help, and where, the likes of VCs are demanding that, you go to people like yourself, Eric.
Yeah, exactly. So I think the time is to first of all give a big clap to Eric and thank you for coming and catalyzing our thoughts. We're now going to move